Firstly, I'd like to start by thanking the organisers for inviting me. And secondly, to say that the talk you're going to hear would have been deemed heretical during the last 30 or so years. Can you hear me all right at the back? Oh, good. Thank you. Well, I'm going to talk about HSV1, herpes simplex virus type 1 in Alzheimer's disease. And you might well ask, why should a virus be involved in Alzheimer's disease, and why this particular virus? Well, in herpes simplex encephalitis, which is a very rare but very serious and horrible disease, the main brain regions affected are the same as those mainly affected in Alzheimer's disease. HSV1 another point is HSV1, once it gets in the body, it can stay there lifelong. And this means that in old age, it could well start misbehaving. It's not like an acute infection where it gets in, does damage, kills a person or doesn't kill them, and then flies out, which is what a lot of people think viruses do, but that's not the case for these viruses which show latency, dormant. The other point is that the virus is very common, as unfortunately as Alzheimer's disease. Oh, not moving on. Oh, it's not moving on.
There we go.
No,
there. Oh, went too far.
This doesn't seem to be working.
Okay.
Is that one?
Yeah.
Okay, thank you very much.
Next one.
No, that's it. Yes, okay, thank you very much. Well, having said that, what we decided to do was to look for viral DNA in brain by PCR. This was way back in 1989 when we started that work. PCR had just come in, and so it was obviously a very good method to see whether or not we could detect any virus DNA in brain of Alzheimer's and elderly age-matched normals. So what we did was we extracted the DNA from frozen post-mortem tissue, and this shows the results. The left-hand side is the prevalence of DNA, HSV1 DNA that we detected. And as you can see, the Alzheimer's and the elderly normals are fairly similar, whereas young normals' brains very much lower prevalence. This was very disappointing because we very naively thought that perhaps the virus might be present in Alzheimer's brain and absent in normals, but how often does one get the answer that one would like or expect? And the answer is not very often. So on the first sight, that seemed really quite disappointing, but we persevered. Sorry. I think I'm sorry, can I just check this? I think I've missed out quite a lot of slides actually here.
Is it this one you're looking for?
Perhaps not. Okay, we'll be on. Okay, that's fine. Okay, thank you, yes. The questions we asked... Sorry. I'm sorry. Yes. Anyway, what's the evidence for viral presence, not just ours, but in brain? We detected the same brain extracts by PCR, and I should point out this was the first microbe shown definitively to be present in elderly, normal, supposedly sterile human brain. Its presence depends on brain region and on age of subject. The prevalence was in our hands about 60 to 70%, but the number of samples was quite small, so I wouldn't take that as very literally exactly correct. I should also point out that HSV1 is the main agent that's been implicated, infectious agent implicated in AD, but other pathogens are probably involved too. But I won't have time to talk about those. Anyway, we then decided that we should look for a host factor, because often the susceptibility to infection or the severity of infection can depend very much on host characteristics, genotype, and so on. And the obvious thing to look for differences between elderly normal brain and Alzheimer brain was the ApoE allele frequency. ApoE is a lipid carrier in the blood, and it was found in the 1990s, yes, to be one of the main risk factors for Alzheimer's disease, ApoE type 4 allele. So we looked at the ApoE genotypes of various of our samples from the Alzheimer patients at age normals, and what we found was very striking. As you can see, there's quite a large association, a high association of, sorry, this has gone on to the next slide. I'm sorry, this seems to be jumping slides, but
This is just at the bottom. If you can.
I don't know what's happening.
That's right. What I can do is take you back, I think,
We've missed out a slide.
to that.
That's
one.
it, go on,
And
thank
then
you.
the pointer is that one there.
Yes, thank you, I know about that. So it's a right-hand one. Okay, thank you. There's a very strong association between the allele frequency, ApoE4, and of HSV1 positive AD patients, but a very low frequency of ApoE4 in HSV1 negative Alzheimer's and in the elderly normal, both HSV positive and HSV negative. And for various arguments which I haven't got time to present now, we suggested that this was probably causal, not just artifactual, not just an association. We then did another very speculative experiment, which I never thought would work, but gratifyingly it did, and that was to look at the ApoE allele frequency of herpes labialis, cold sores. The reason we did this was herpes labialis is known to be caused usually by HSV1, sometimes by HSV2, genital herpes, but normally HSV1. And so it seemed interesting to find out whether there was any possible association of ApoE4 allele frequency. And to our surprise and perhaps delight, we found this was indeed the case. Here's the allele frequency of the cold sore sufferers on the left, and one of non-cold sore sufferers, who actually the majority, it was only about something like 20 to 40% of people who are infected with HSV1 actually develop cold sores. So the rest are asymptomatic. So just to summarize those results, HSV1 DNA is present latently in many elderly normal Alzheimer brains. They say it was the first microbe to be shown present, which caused an awful lot of shock and horror by people who wouldn't accept that, and our paper consequently took ages to get published, and it also affected very much our ability to get grant funding because we were so heretical. Then there seemed to be evidence that it's latent in brain normally, but it reactivates probably recurrently. And HSV1 in brain of ApoE4 carriers confers a strong risk of Alzheimer's, accounting in this small sample for about 60% of patients. And ApoE4 is a risk factor for herpes labialis. So conclusion was in the CNS and PNS, there was probably greater HSV1 damage, or perhaps lesser repair, in ApoE4 carriers. And one point I'd like to make is that neither having Alzheimer's disease nor carrying an ApoE4 allele predisposes to HSV1 infection of the CNS, because those are possible other alternative, more trivial explanations. I'd just like to digress for a moment to mention that we decided to look at this point, go off the Alzheimer's story and look whether ApoE had any effect on outcome of various other illnesses caused by infections. And just to show you the most striking and most convincing, we found that hepatitis C induced viral, hepatitis C virus induced liver damage. In that case, ApoE4 was strongly protective, and that of course caused quite a lot of astonishment and outcry as well, because that went against the idea that ApoE4 was the baddie. but in fact it turned out here to be quite strongly protective. We then decided to look for whether or not there's any link between HSV1 infection and the characteristic features of Alzheimer's disease. And what we did was we infected human neural type cells, glioblastoma, or in this case, glioblastoma cells, and we looked for evidence of A-beta by staining. These top row are all infected, and these are uninfected at the bottom. This, by the way, is infected mouse brain. And you see for A-beta here, beta amyloid, which is the main component of amyloid plaques in Alzheimer's disease, there's quite strong staining after infection. Same is true in mouse brain. And in this case, it shows staining for abnormally phosphorylated tau, which is the main feature of neurofibrillary tangles, another major feature of Alzheimer's disease brain. So, yes, it does produce the main characteristic pathological features of disease. So that linked quite nicely. But that again produced a lot of shock and horror, and it took ages to get that paper published. Anyway, move on to the next. So to summarize those, HSV1 infection of human neural cells in culture increases intracellular A-beta, two types of beta amyloid, and levels of the enzymes responsible for creating the A-beta, and tau phosphorylation, which we checked at various phosphorylation sites and found the same answer for each of them, increases also. And these results have since been confirmed and extended by a number of other labs. And also brains with HSV1 infected mice show A-beta reposition. We then did another speculative experiment. I think really I was taking quite a lot of risks, but again, fortunately, this did work out. And this was really extraordinarily surprising and really very, very gratifying. We did in situ PCR, which is a very difficult technique and enables one to look at tissue sections. And what we did, we stained for viral DNA and we stained for amyloid. And the first one is HSV1 DNA stained after in situ PCR. And this one it's stained, the tissue section has been stained for plaques. And then the two merge, and as you can see, the colors have come out rather oddly, but anyway, you can see they merge quite very neatly indeed. This is another section, and here the only difference is that the beta amyloid was stained by immunohistochemistry. I'm afraid you probably can't see the colors, but one's purple, the HSV1 DNA is brown in both cases, and the other A-beta is purple, but I can assure you it's a complete co-localization there. That created a lot of interest, which helped our cause, really. We found that most of the DNA is present in AD plaques, only a much lower proportion in aged normals, and nearly all the plaques in both cases contain viral DNA. So this is just to summarize really what I've just said. Most of the amyloid plaques in AD brain contain viral DNA. And we asked the question then, is the virus encaged by the amyloid? Is it acting sort of protectively? And we did try some experiments to test that. But unfortunately, you had to buy. We couldn't afford to make the amyloid, so we bought it. And unfortunately, it did show, although it showed protection, it also was fatal for the cells eventually, so we had to discard the results, unfortunately. Anyway, as I said, most of the DNA, viral DNA, is located in plaques in Alzheimer brain. And we could refute artifactual get back. we could refute the possibility of co-localization, which some people suggested was actually happening. It does really occur. Where's the... So association does not prove causality, but as HSV1 infection causes A-beta accumulation, as we showed earlier, that and the co-localization of the viral DNA and amyloid indicate that HSV1 is a major cause of the toxic A-beta products and the plaques. This was one of the missing slides, which somehow got lost. I don't know why. I asked 4 questions, and these are the answers I found. The questions are obvious. Is virus present in normal brains? The answer is yes in the elderly. It's been shown we showed it by solution hybridization and by in situ PCR. And is it active in brain? Yes. We showed that. I haven't showed the slide, but we found that. And it's probably recurrent, which we showed by detecting intrathecal antibodies to HSV1 in the CSF in the elderly, and hence it's likely to cause damage. And what other factors link HSV1 with AD? ApoE4 carriage accounts for about 60% of the cases, and ApoE4 is a risk for cold sores. And is there a causal link with AD neuropathology? Yes, because we showed that HSV1 infected cells and infected mouse brains produce A-beta. So this suggests that, sorry, the pointer seems to have gone, but never mind. This suggests ApoE4 carriers suffer greater HSV1 damage, and also suggests the possibility of antiviral treatment. And that's what we looked at next in cell cultures. We use valacyclovir, which is a product of which is used to treat patients with HSE, and but anyway, we use acyclovir in cell culture, and what we have here is increasing levels of, as you go down, increasing levels of the acyclovir, HSV1 infected on the left, and A-beta stained, uninfected on the right, and you can see a big difference between the two. Staining, a lot of staining on the left, no staining, or virtually none in the uninfected, and the same increasing valacyclovir concentration as you go down, HSV1 infected with heavy staining, particularly at the beginning, and gradually decreasing staining with increasing concentration of the acyclovir, and then the uninfected, very little staining indeed. This is just a plot showing how this, the levels of both A-beta and p-tau varied, and HSV1 proteins. And the top graph shows a decrease of the viral protein almost to about 40%. This is quite explicable. I won't explain it now, but there are reasons why it doesn't go down to 0. A-beta, rather similar finding, that decreases with increasing acyclovir concentration down to about 30%. But in the case of phospho-tau, it decreases almost to 0 with increasing acyclovir. This actually reflects the stage in the viral cell cycle where it's actually, these proteins are actually synthesized. So this really spoke strongly in favor of antiviral treatment for Alzheimer's patients. So overall summary, viral DNA resides latently but reactivates in brain of many elderly people. Virus in brain and ApoE4 confer a strong risk of AD. HSV1 infection of human cells causes AD-like changes, and the viral DNA is located within AD plaques. Antiviral treatment reduces greatly the production of A-beta and p-tau. And something I didn't mention before, I did a few experiments with a colleague in Sheffield, vaccination of mice with mixed viral glycoproteins, and we found that actually prevented, in infected mice, establishment of latency in brain. And then there was the other thing, not a non-Alzheimer study, where we found that ApoE determines the severity of damage or susceptibility of infection to infection by diverse pathogens, which by the way, we interpreted as being which allele is, or which isoform competes of the ApoE protein competes most successfully with ApoE to allowing or not allowing viral entry into the cells. But that hasn't been, we haven't been able to follow that up, unfortunately. So this is what we formulated, and I'll leave the rest of the talk, assuming there's time, show how we tested or went into this. HSV1 resides latently, we reckoned, in brain of many elderly people, both Alzheimer's and elderly normals, but can reactivate probably repeatedly. And that causes damage which must be limited and localized, otherwise they would show the symptoms of HSE, but cumulative, and it leads eventually, we suggest, to AD. And in the brain, in combination with a genetic factor, ApoE4 confers a strong risk, probably more damage in ApoE4 carriers. And ApoE4 is a risk for herpes labialis, and antivirals protect against HSV1 damage. And there are now about 500 studies using very diverse approaches which support HSV1's role in AD. As I said, there are other infectious agents which are probably involved as well, but I can't talk about those. So what questions did we ask after this? Well, firstly, is there evidence of HSV1 reactivation in brain? And if so, what causes it? And is there evidence that HSV1 reactivations lead to AD? I mean, these are fairly critical points arising from what our concept is. And so we tried to test what we could of them. Well, firstly, what causes HSV1 reactivation in life? There are many types of injury that do. Infections, for example, COVID, inflammation, hypothermia, immunosuppression, axonal damage, which might be caused by brain surgery, and concussive impact, traumatic brain injury, all cause reactivation. And what's the evidence of reactivation in the living brain? Well, I mentioned before, HSV1 antibodies are present in the CSF of Alzheimer patients and of normals. And in other words, the DNA, viral DNA, replicates and is expressed in brain. HSV1 is found, well, I won't go through all of these, but it takes too long, but I should stress an important series of studies being carried out by a group in Rome showing that in infected mouse brain, a repeated reactivation by hypothermia of HSV1 in the infected mouse brain, where it's latent, causes AD-like phenotype, cognitive decline and synaptic dysfunction and various other things. So in mouse studies, this sort of reactivation leading eventually to Alzheimer's has been shown. I won't go into that. This showing you we did some studies very early on by in situ hybridization, which pre-PCR, that was in 1986, really a long time ago, but I haven't got time to go into. But it shows that immunosuppression does allow detection of what must be reactivated HSV1 in human brain. In people who weren't immunosuppressed as part of their treatment, there was no sign of any DNA. Presumably not enough had been reactivated, or none had been reactivated, and nothing therefore replicated to cause an amplification of the DNA. So I'll just briefly say that evidence of reactivated HSV1 involvement has been shown by epidemiological studies and genetic ones. Things like, for example, the second point here, dementia risk is greater in HSV1 infected people than uninfected, and antivirals decrease the risk. Genetic support for a viral role, linked pathways found. I should say not everybody has found the same epidemiological effects, but I think that's partly due to the fact that a lot of people don't seem to realize that people who are asymptomatic shouldn't be, but who are infected, should not be classed amongst controls, yet a surprising number of people have actually done that. They've included in their controls the asymptomatic virus-infected people, which really seems very strange to me, because obviously they should be in the infected class.
We probably need to wrap up. If That's okay, sorry, we need
Oh,
to
sorry.
move on to the next speaker.
Oh, sorry, yes, okay.
Thanks.
Well, we showed by 3D brain model that HSV1 infected people. This was shown by a group at Tufts, and so this was very strong support for the HSV1 infection idea of Alzheimer's. They developed Alzheimer's like phenotype. And we used the brain model to show that two major risks for Alzheimer's: infections and head injury cause reactivation of HSV1. I won't go into that, of course, no time. So we conclude that in 3D brain models with latent HSV1, infection and traumatic brain injury reactivate HSV1. And in latently infected mice, hypothermia reactivates virus, leading to AD-like phenotypes. And just to skip the next one, infection and other types of injury can reactivate virus, and also HSV1 infected people are higher risk than uninfected. And all these suggest that reactivation of HSV1 in brain increases the risk of Alzheimer's, and some viruses reduce the risk. So we propose that after injury of these types, if not others, reactivated HSV1 in brain leads to the development of Alzheimer's dementia, and HSV1 is a major cause of the disease. Thank you. I'm sorry I overran myself.